Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. Dear students, today we are going to discuss about double helical structure of DNA, or we can say Watson and Crick model of DNA. Before this, we have discussed about proteins, we have discussed about carbohydrates. Now, today we are going to discuss about DNA, its double helical structure. In this topic, in nucleic acids, we have discussed about DNA in general. Now let us have a look on what is Watson and Crick model of DNA. In 1953, J.D. Watson and F.H.C. Crick proposed double helical structure of DNA molecule. Watson and Crick proposed the convincing model with full explanation of the arrangement of molecules in DNA. In summary, the silent features of the Watson and Crick model of double helical structure of DNA are as follows. DNA is made up of two long chains. Each chain is made up of many nucleotide units. Hence, each chain is called polynucleotide chain. Let us have a look on it. This is DNA double helical structure, or double helical structure of DNA, or we can say Watson and Crick model of DNA. This is DNA which is made up of two long chains. These are called polynucleotide chains. Then I can say that these polynucleotide chains are separately coiled to form double helix, right-handed helix about central axis. These polynucleotide chains, these are separately coiled and they form double helix. Two chains of DNA are complementary to each other. If the sequence of bases in one chain is AG, AT, GC, the sequence of bases in the other chain is TC, TA, CG. Means if the sequence of, here I am going to show you this, if the sequence of one strand, one chain is adenine, guanine, adenine, thymine, guanine, cytosine. The sequence of bases in the other chain is thymine, cytosine, adenine, cytosine, guanine. Because adenine always base pairs with thymine, G with guanine with cytosine, and so on. The two polynucleotide chains are antiparallel. That is, one strand has phosphodiester linkage in 3' 5' direction, while other strand has phosphodiester linkage in just reverse, or we can say 5', 3' direction. Let us have a look here. This moves in 3', 5' direction, and this one moves in 5', 3' direction. So these are antiparallel polynucleotide chains. Each polynucleotide chain consists of alternate sugar phosphate backbone laying on the outer side of helix. Let's have a look. Each polynucleotide chain consists of alternate sugar phosphate backbone. This is backbone of polynucleotide chain, which is made up of sugar and phosphate molecules on the outer side. The purine residues, the purines and pyrimidines, the purine bases and the pyrimidines. Purine bases include adenine, guanine. Pyrimidines include cytosine, thymine. Once again, purine bases include adenine and guanine. Pyrimidine bases include cytosine and thymine, or stacked one above the other in the interior of double helix. These are stacked one above the other in the interior of double helix, nearly perpendicular to the long axis of helix. Distance between the two successive with a two successive base pairs is 3.4. Look at here. Distance between two successive bases is 3.4 angstrom, and one complete turn of helix with pitch of 34 angstrom, and this complete turn of pitch, which is about 34 angstrom, Each turn of helix consists of about 10 base pairs. In each helix there are 10 base pairs. And let us have a look on the diameter. Diameter is 20 angstrom. One of the major folding force that holds the two chains together is hydrogen bonds between base pairs. Thymine is always base paired with adenine by two hydrogen bonds, and cytosine is always base paired with guanine by three hydrogen bonds. Let us have a look here. This adenine, this is base paired with thymine with two hydrogen bonds, and this is here thymine, adenine, and guanine is base paired with cytosine with three hydrogen bonds. So we can say that DNA molecule of double helix looks like a ladder. The sugar and phosphate or phosphoric acid form the backbone, and base pairs forms horizontal rungs. So this is like a ladder. You can see this forms the backbone. This forms the backbone, sugar and phosphate, and these base pairs, they form horizontal rungs. It is all about structure. You can also have a look here. This is sugar, deoxyribosugar, sugar, phosphate, adenine, and this moves in this direction in one strand, and it will move in opposite direction in another strand. Have a look here. Here also. Once again, these are separately coiled two polynucleotide chains. Their backbone is made up of sugars and phosphate, and from external side and their internal side, sugar and phosphate. From internal side there are base pairs. There are adenine, AT, GC base pairs. We can say their bases are stacked one above the other in the interior of double helix. So there is also two successive base pairs is about 3.4 angstrom away from one another, and one turn it is about 34 angstrom, and there are about 10 base pairs in one turn. Look, it is diameter. It is here diameter. It is 20 angstrom, and these are anti-parallel. Means one strand, if it moves in 3 to 5 direction, and other moves in 5 to 3 direction. And these are also complementary strands.